Good morning, friends. Today we have to see the new topic. In the last lecture, we talk about the detector, UV visible detector. In that, we have seen their diagram. Then we talk about the construction and the working, and in the advantages of this particular UV visible detector. This is all about the instrumentation part of the HPLC, that is a high performance liquid chromatography. As already we have seen the different components of this HPLC. In that the detector is one of the most important thing which helps in that particular quantitative and the qualitative, or that we can say the analysis. Now we are going to discuss, we have to see the new second type of the detector, that is a refractive index detector. In the UV visible detector, we have seen that depends upon the UV source. That we have seen the mercury, that was the low power mercury was there, that we have seen low pressure mercury lamp is there, that can be used as a source for the UV visible detector. Here in case of the refractive index, you know refractive index very well. It is also called as the differential refractive index defector. It is also called as the differential refractive index detector. The detector are based on the changes in the refractive index of the solvent, that is caused by the analyte. In this detector, the solvent passes through one half of the cell to the column. Then dilute air flows through the other chamber. Here you can see the diagram is there, mirror is there, sample cell is there, okay, and then the lens is there. This very simple diagram is there. A refraction should be occurred. You can see light is passed through this lens to this sample, then reference is again there, then the refraction is there. That light, whatever the light refracted, that will be sent to the that your amplifier. Here the zero adjustment is there, then amplifier or recorder. These things are there. This is a general diagram of this particular differential refractive index detector. In that, what is that, we can see this is based on the changes in the refractive index of the solvent, which is caused by the analyte. In this detector, the solvent passes through one half of the cell to the column. The dilute air flows through the other chamber. The two compartments are separated by the glass plate mounted at an angle such that bending of the incident beam occurs if the two solutions differ in room temperature. The resulting displacement of the beam with respect to the photosensitive surface of a detector causes the variation in the output signal, which when amplified and recorded provides the chromatogram. The detector responds to all the solute. They are reliable and unaffected by the flow rate. They are sensitive to high temperature, must be maintained at a constant temperature. They are not sensitive as other types of detector. The limit of the detection for this detector is, it is given 1000 nanogram to 1 milligram. Okay, this is the detection limit. In the milligram of the milligram, you can detect milligram to nanogram. That is the limit of this particular. What is there? Basically, you can see in the diagram that what is there. You can have this particular detector is based on the changes in the refractive index of the solvent. How the refractive index will change? Refractive index will change by The analyte, that is whatever the present in the particular that thing. Okay, means if we say in the solvent there is analyte. Okay, that sample is there. What are the sample molecules are there? They may lead to cause the change in refractive index. In this particular detector, the solvent passes through one half of the cell to the column. The elute air flows through the other chamber. That is very much important. What is there? Solvent passes through one half of the cell to the column. The elute air flows through the other chamber. Here you can see the sample and the reference material is there. From one chamber it is going to pass and another and eluted from the another chamber, that is there. It is very much means one one path is for the entrance and another for path is for the exit. The two compartments are separated by the glass plate mounted at an angle such that bending of the incident beam occurs if the two solutions differ in the voltage. That is very much important. Then and only the refractive index will be there. Once there should be the refraction, and that's why you have to separate that compartments by the glass, which should be mounted at such an angle which can help in the bending of the incident light. The resulting displacement of the beam with respect to the photosensitive surface of a detector causes the variation in the output signal, which when amplified and recorded provides the chromatogram. Then it will gives you the chromatogram. How the chromatogram will be get? Whatever the signal is coming from that particular detector is sent to the amplifier. Amplifier, the role of the amplifier is to amplify the signal, to increase the strength or the intensity of the signal, and then it will send to the recorder, and recorder will give you the graph, that is nothing but the chromatogram. Are you getting that point? Then what are the advantages of this particular refractive index detector? It should have to respond to all the solid. It is responding to the all the solid. They are reliable and unaffected by the flow rate. It is the second advantage of this particular detector. Then next, they are sensitive to high temperature and must be maintained at constant temperature. It is again very much important. What is that? The third most point is that they are sensitive to high temperature and maintained at a constant temperature. They are not as sensitive as other types of detector. It is not that much sensitive as other type of the detector. And limit of the detection for this detector is 1000 nanogram to 1 milligram. That is, you can use that particular detector for the detection of this concentration. Very simple. It is again two types of the detector is there in the exam. Sometimes that one of that detector is there, but you have to read all these things because the exam pattern may vary. If the multiple choice questions are there, then you have to read all the part very well. Please make the habit of the reading daily the whatever the given notes to you, because MCQs will be from that particular notes only or that material only, because all the part is very much important as far as the year that exam is concerned. Very simple that description is there for this particular refractive index detector. Main thing is that the principle of this particular refraction should be there, and refraction it will ultimately gives you the refractive index. Okay, which thing will responsible to get the refraction, that refractive index? That is solvent will be responsible to get the refractive index, that you have to remember. Okay, here this is the that detector which is used in case of the HPLC, that is a high performance liquid chromatography. You have to remember these things in particular HPLC, which is the that is a mobile phase and what is the stationary phase. That things we have to remember. Then and then only this chromatography technique you can understood very well. Please make it clear that there should be always a reading, because reading makes perfect. Okay, you have to read that part very well. After reading, you have to remember these things on your own, because in the exam you have to appear, you have to write the answers for the questions. For this purpose, please go through these notes well. If you have any doubt, any query, any problem, you can ask it on the Google Classroom, or you can ask it on the WhatsApp group. Please be free to ask the queries whatever you have. Okay, please complete all these things in time and make the habit of reading, because reading makes an perfect. Here we will stop. Thank you.